Got that recording. Okay, so what we're going to do is we're going to start with the first sort of topic in physics. It's a bit of a strange topic, to be fair. It's sort of a bit of everything in terms of units and prefixes and measurements and errors. So it probably won't take us the whole session to get through this, and then we'll probably start the next topic as well. What I will do is I will send over some sort of work to go with these units, because it's lots of sort of skills of converting units and standard form and prefixes, things that aren't completely new, things that will have been addressed in GCSE physics, actually in GCSE science in general, and probably actually in maths as well. So it's sort of a recap with a few new things in. And then, yeah, it's just sort of getting used to using the maths behind it. So actually what was probably easier for you is then once we've done this, is if I send through the slides and then some questions about them, and you can have a go at working through them. And then if there is any issues, you can ask them about them then. But yeah, so SI units. So these are standard scientific units that we use in science. Now, okay, so we basically split out SI units into base units and combined SI units. And so our base units are like our pure, original, basic units that we can't change and have any different versions of. They're almost like as if we were talking about primary colours, but we're talking about units instead. So this is just going to literally be me listing through a few of them, and I'll send them all over to you in a bit. So in SI units, our base unit for length and distance is going to be meters. And what we mean by this is our standard unit. We would automatically always defer to using meters. Hence why when we do things like speed, it's usually meters per second. And then if we're talking about volumes and densities or areas, we're going to want to refer to meters. We can still use centimeters, kilometers, and all the other variants, but this is generally our one that we would defer to straight away, and that's going to be the same for all of these. So there'll be one that we like to defer to every time. So our base unit for length and distance is going to be meters. And electric current, our base unit for electrical current is ampere, or also shortened to amps. There is other versions and other ways we can calculate electric current, but this is our base unit for electric current is amps. Then we'll have time. Our base unit for time is usually seconds. Again, that's one that you'll start to see when velocity and speed and acceleration, if we're talking about meters per second. Seconds, the one we'll defer to most of the time. Again, we can use hours, we can use milliseconds, we can use minutes, but it will generally be seconds. Mass, our base unit for mass is kilograms. Generally, just because most of the time when we talk about science, because we're doing things ranging from biology to physics, we've got things that are super tiny, things that are super huge. So we've sort of gone with, okay, we'll go with kilograms, basically. That's sort of in the middle. And then we use standard form to make it a little bit more clearer. And then we have a base unit for chemical mass. Now, chemical mass is something we've actually already touched on when we were talking about chemistry the other day. It's going to be the mole. And we'll look at the mole a bit more next week. And actually, because we've decided to use kilograms for mass in general, and kilograms is actually quite a large mass, it's quite a large quantity. When we're then going to talk about chemicals and elements and atoms, using kilograms is really an inefficient way to talk about them. So that's how we get that idea of the mole again. And the mole is a certain amount of stuff, and moles can literally just be a certain amount of stuff. So we have Avogadro's constant, which is our amount of stuff per mole. So we could have Avogadro's constant, and we're talking about, I have a mole of feathers. So mole we can use for anything, and we use it for chemical mass because it's nice and easier than trying to talk about kilograms, because we would have such tiny numbers. And we have a base unit for luminosity intensity, and this might be one that you haven't covered yet, and this is our candela. So we just sort of have to know these at the minute. It's a bit of a strange topic. We're just sort of listing units and knowing them. And we have our base unit for temperature. Now, our base unit for temperature is Kelvin. We can still use degrees Celsius, but we can convert Kelvin into degrees Celsius. But from this point, our base unit for temperature will always be Kelvin. So then we have combined SI units. So this is where we're maybe taking a couple of our base units and we're putting them together to get a combined SI unit. So for example, speed with our meters per second. So we're using our base units for distance and our base units for time to get meters per second. Acceleration, meters per second squared. Momentum, we're going to use kilograms per, kilogram meter per second. So we're combining quite a few base units there. And then like electric charge. So we've already mentioned electric charge and said electric current, sorry. So electric current, we use ampere. Electric charge, we're going to use a coulomb, which again is possibly a new unit that you haven't come across yet. When you start seeing the C on its own, and that's a coulomb, and we'll look at that when we get onto the electricity topic. Potential difference, this is one that you will have done before. Potential difference is our volt. Force, a Newton. And again, these are classed as combined units because force is something that's working with, it's a combination of some of those other units together. So whilst we talk about it in the Newton, it is a combination of some of those base units. Energy is measured in joules, and our electrical resistance is in ohms. Again, this is just a bit of a strange list. Temperature, we can measure temperature in Celsius, in degrees Celsius. To be able to do this, we have to convert degrees Celsius, Kelvin, sorry, into degrees Celsius. So all we have to do is we would take whatever the Kelvin measurement is for the temperature, take away 273 from that, and that would give us a temperature in degrees Celsius. That is a useful conversion to know and to remember because it can come up quite a bit. And because Kelvin is a base unit, what can sometimes happen is you'll get a question or you'll get some readings and you've measured the temperature in degrees Celsius, but then to do a further calculation further on, you'll need that temperature actually in Kelvin. So being able to convert between degrees Celsius and Kelvin is quite useful. Obviously, if we just need to do it the other way around, rather than taking 273 away from the Kelvin reading, we'd do the opposite and we would add 273 to our degrees Celsius reading. Pressure is in pascals. You may have come across this before, you may not have done, you may have had some other variant of pressure units being used for GCSE, but at this point now we use Pascal for pressure. Hertz is our frequency. You may have come across this one when talking about sound, ear and wavelengths. And magnetic flux density, again, this could be a new one, and that's Tesla. And we'll come across that again later on. It's sort of a, we're going to talk about it now and then come back to it. So magnetic flux density. And then finally power, which we can measure in watt, which shouldn't be a new unit. So then we sort of come on to prefixes. And prefixes we've actually spoken about a lot. We use prefixes to basically indicate the scale of a value. So in a way, when we talk about biology and chemistry, when we have our di, tri, and so forth, we also have prefixes in maths and physics, which obviously we use these as well in chemistry and biology. And these indicate the scale of our values. So these are some that basically it's just really worth knowing, and learning. Just because you can get asked to convert between different units. So it could be convert something that's in kilograms into picograms, convert something that's in nanograms into micrograms. So you have to be able to do those conversions. So knowing the prefix, the symbol, and what it's multiplied by is really useful. I won't sit here and read through them all. The ones that will come up a lot Our femtos, when we're talking about atoms and subatomic particles, that will come up a lot. Your picos, your nanos, your micros going to come up a lot again when we're talking about chemistry, when we're talking about biology and biology in the lab, when you're carrying out actual experiments and biological experiments with things like DNA. The units that we'll be using would be pico, nano, micro, but no smaller than that. Milli, which you'll come across a lot. And then obviously we get into the kilo, mega, giga, tera, and peta, which are generally, once we get past kilo, your physics realms. We don't use them too much in chemistry or biology, so this is a nice physics area that we'll start to use these larger numbers as well. So physics, we sort of use this whole range of mathematical values, so it's really quite nice. So yeah, so we have to know our prefixes as well, and I'll send some stuff over about calculating, converting between different prefixes. Obviously, rather than writing a massive number out, we have these multipliers, which are part of standard form. And obviously you'll know standard form. I'll talk it through with you again anyway, but standard form is nice because rather than having this either huge number with loads of zeros written down or this tiny number with loads of zeros written down, we can put something into standard form. So standard form. Our general equation to say for standard form is A times 10 to the power of n. A is our number that will be between the value of 1 and 10. We will never have anything above 10. People generally like to use a number between 1 and 9 rather than 1 and 10, just because obviously then if we can get that 10 in there, people would put a decimal place in. And so it's usually a number between 1 and 9, but officially on the mark schemes and the exam board it is a number between 1 and 10, but you'll usually find it's a number between 1 and 9. And n is how many places to move our decimal point. So you can get asked to write a number in standard form quite a lot in physics. It's a lot of test of your math skills and understanding standard form, understanding what it means. So getting to grips with standard form early on is really important, because actually we're going to learn what these sizes of numbers actually represent. Because what we can start to do is we can start to sometimes forget how big or how small a number is when we don't start looking at these times 10 to the power of whatever. If we don't start looking at those, we can sort of forget because we're just dealing with 1s, 2s, 3s, 4s. So for example, you can get asked, write 346,000 in standard form. So there is a process to use. You probably know how to do this anyway, but I'll talk it through as we go. So first thing, you're going to put in your decimal place so that you have, like we had our A number is a number between 1 and 10, 1 and 9. So we'll make it 3.46. Then your second stage is you're going to count your decimal point moves. So from where it was originally to where you've ended it up in 3.46. That was that was not 10 decimal point moves. It was 5 decimal point moves. So we end up with 3.46 times 10 to the power of 5. And it's that simple. We're just moving it along and we're going to count them each time. Converting units. This is another thing you have to be able to do. And so that's something I mentioned back when we were talking about prefixes. So converting units can be something nice and simple. For example, meters to kilometers. So we're saying in the same realm of the unit, we're just basically actually changing its prefix. We're changing its power of 10 almost. So what we want to do first is want to work out what we're converting to what. So we're going meters to kilometers. So then you need to come up with your conversion factor. So for meters to kilometers, it's really easy. We know 1 kilometer divided by 1,000 meters is our conversion factor. So we've got 1,000 meters in 1 kilometer. So then all you need to do is 80 times it by your conversion factor. That would give you 0.08 kilometers. So that's quite nice and simple, but you need to know your conversion factor. So this is why I say learning the prefixes is important, because actually we need to know our conversion factors. So you need to know how much bigger or smaller a picogram is to a milligram. and how much a decigram is to a milligram. So it's knowing those sorts of pieces of information that can't really get taught as such. It's sort of just a learning. It's a bit of a slow process of just keep going over it. But it is really important because obviously, as you know, when we're doing any sort of calculations, everything needs to be in the same units. So you might get a question where, for example, part of the question is in meters, part of the question is in kilometers. You need to make sure they're in the same units. It regularly comes up in things like biology, where in real life, it often doesn't happen in the exams, but in real life, you'll be dealing with something that's in nano and micrograms. So one thing that you're measuring is in nano, one thing you're measuring is micro. But actually, if you're then going to do a calculation, you need to make sure, convert them both into the same unit. Otherwise, your answers are wrong. It's something that will come up a lot in microscopy as well. So it's just making sure we know that we're always using the same unit. So we need to know our conversion factors. Some of them are really nice and simple. An example of a slightly more complicated one is 2 joules to our electric volts. So joules to EV, we would get our conversion factor. This is one that you would want to learn. So that is 6.2 times 10 to the power of 18 over 1 joule. Don't know why my joule's got a little extra line above it. Just ignore that, pretend it's not there. So then we would do, so that is for conversion factor for 1 joule. We obviously want to do 2 joules, so we would do 2 times 6.2 times 10 to the power of 18, and that would give us 1.2 times 10 to the power of 19. I just haven't made my 19 small, which is a little bit annoying. So that's what you'd get there. So that's a conversion factor that you would want to learn. Most of them, again, you're going to want to learn all of them, but that's one that you need to sort of know off by heart almost. So that's sort of the first bit of units and measurements. It's a bit strange. We sort of just have to know them. And then we look at limitation of physical measurements. So we've spoken about what we're going to measure things in, what units we're going to use. But what actually is our limitation of physical measurements? There is lots of measurements, and there's lots of physics sort of gets divided into theoretical and physical physics. That's the word I was looking for. And so we can either do theoretical, mathematics, or we can actually do research, and we can actually carry it out with physical measurements. But we obviously have to understand that there is a limitation to our physical measurements, and we have to take into account some of those issues that we have. Lots of this, again, probably will be quite similar to what you've covered at GCSE, because I know you did really well at GCSE. We didn't used to cover it as much, so it's all this sort of precision, accuracy, reproducibility, but we'll go over it again. Some of the definitions are slightly different now that it's A level, but it's quite good. That gap has been bridged a little bit better than it used to be. So we have limitation of physical measurements, and what happens is here is we're going to look at physical measurement errors, and we have something called a measurement model. And what we have here is we have the observed value, so that is a measurement you've made. So for example, you've gone and you've measured the ionization energy, first ionization energy of nitrogen, and that would be your observed value. We would also have a true value, which is what the measurement should be. And because this is an experiment that's already been done before, we can compare the two values. We know what our measurement is, and we know what it should be, and they might not match. So this could be a physical measurement error that could come into place. So we get something called an uncertainty error, and this can happen a lot. So what we basically do is to work out our uncertainty error, we have our observed value, and it will always equal our true value plus our error, because if there is an error that's come into place, if our observed value does not match identical to our true value, that means there's been some form of error. So our uncertainty, our error, is our observed value equaling our true value plus our error. Now, our error can be due to two things. It can be due to either a random error or a systematic error, or it can be both. So technically, our uncertainty equation for our uncertainty can be our observed value equals our true value plus random error plus systematic error. There's probably always going to be some random error that comes into play. Systematic error, not necessarily, but that is the full equation for it. So we're going to have a little quick look at systematic error. So systematic errors, these are errors that are going to follow a set pattern. They are really, really hard to avoid. The main ways of avoiding them is making sure that we're using measuring equipment and that we calibrate equipment. So for example, calibration is really, really important for all pieces of equipment and apparatus. So for example, if you're using scales, a balance, weigh something, measures the mass of something that you know already what its mass is. So for example, you might have a 50-gram loaf of bread. I feel like that might be a little bit ridiculous, but we'll go with that. We'll go with 50-gram loaf of bread. You know that weighs 50 grams. You know its mass is 50 grams. You put it on your balance, you read the measurement, and it doesn't come out at 50. So you know there's a calibration error. So then you can calibrate your scales to then reset it again. And so calibration is really important. And in big places like labs, where equipment's being used a lot, calibration has to happen regularly. And you will have basically a set of equipment that you would calibrate everything to, because otherwise what can end up happening is you go, oh, if I calibrated A with B and then I calibrated B with C, you can end up still with then the systematic errors coming in. So you'd have like one set of equipment that we just would use for calibrating against. Another really good example is for using pipettes. So in biology and chemistry, we regularly use micropipettes. So there's something that I'll show you at some point, and they're very different to like those plastic see-through pipettes that you've probably used at school. And these are ones that we can set to have a certain amount of a certain volume of different fluids being taken up into the pipette, and then we would pipette it into a different dish. Because we can be working on such small quantities, so we can be working in micrograms, microliters, really, really tiny amounts, it's really important that it's calibrated, because it being even slightly off will change every single sort of result that you're looking at. So they're quite good to be able to calibrate, because in a way, what you would basically do is you already have a volume of liquid, and you know what's in that volume. You know the volume of that liquid that's in front of you. You've used it on other equipment that's been calibrated, you know it works, and then you use your pipette to check it. If there is liquid still left in the original test tube, for example, then you know that it isn't calibrated correctly. If you are pipetting it out and then there's a large air bubble again inside your pipette, you know it hasn't been calibrated correctly. So that's the sort of idea that we go with systematic errors. They are hard to avoid, but the main thing is that we use correct measuring equipment, we calibrate that equipment, and we always try and use the same apparatus. If we're swapping between brands and makes, there can be calibration errors, and so you can end up with systematic errors come into place. Easier ones to avoid are our random errors. Random errors follow no set pattern. They're often due to reading equipment wrong. It's often what people would call human error. What's quite good about random errors is if we are taking a large enough sample size, we've got a large enough data set, what should happen is they should start to cancel each other out. So we will have anomalous results. We will have a random error that's appeared from somewhere because we've read the equipment slightly wrong. Maybe if we're taking the mass of a chemical and it's really, really small, and we're sort of measuring it in picograms or micrograms and nanograms, any just slight gust of wind or move, or if we haven't calibrated it properly with the dish that it's on, can really change it. But if we keep doing this over and over again, what should happen is those results should cancel each other out, any of those random errors. So repeats reduce random errors. So it's why repeats are that thing that everybody always goes on about. We must do repeats, we must do repeats, we must do repeats, because basically not only does it mean that we can spot a random error easily. Also means that it will cancel out. If we have a data set of 100 and we only have one random error in there, it will get cancelled out by the 99 other sets of results that are correct. And then we get to these lovely definitions that you've had at GCSE before. So we talk about accuracy. So accuracy is how close we are to the correct value. So accuracy can only happen if you know what you're looking for, which is a bit of a strange thing. So if you're doing an experiment and you have no clue what results you're looking for, it's very hard to be accurate. When you then repeat it, then you can be accurate, because actually if your results then aren't coming up the same, there's something that's affecting your accuracy in that experiment. But actually accuracy as itself, if we're doing it for a novel experiment, is really, really hard. Accuracy is just how close you are to the correct value. So are we getting that correct value that we're expecting? Precision can get easily mixed up with accuracy. Now, precision is how close to each other our repeated measurements are, and this is independent of the true value. So, for example, we could be looking for the ionization energy of nitrogen again. You've done it 20 times. You've got precise results. All your results are with plus minus, so an acceptable range of each other. They're all of it very close to each other in an acceptable range. However, you can be precise and not accurate. Your results can all be close together, but actually they're not anywhere near that true value. Maybe there's been a systematic error somewhere, your calibration's been wrong, so it hasn't met up. Your repeatability. This again can get confused with reproducibility. So repeatability is the same person doing the experiment with the same equipment, the same apparatus, and getting the same results. So this is basically your repeats. Can you keep repeating this experiment yourself and with the same equipment, and can you get the same results? The difference with reproducibility is can somebody else perform the same experiment with the same apparatus, with the same equipment, and get the same results. So there's just that very slight difference. Repeatability is you being able to do it again and again, and reproducibility is somebody else being able to do it again, getting the same results using the same equipment, but it's somebody else. So you're almost taking out that human error, that random error aspect. And the last little bit is resolution. So that is our maximum precision. So we want to have as high a resolution as possible. We want to be as precise as possible. So when we talk about resolution, we'll often talk about having a high enough resolution for our, if we use the pipette for an example, if you're trying to pipette 0.1 microliters, you are not going to want to use a pipette that is in milliliters, for example. It's not going to be precise enough. You're probably actually not going to want to use a pipette that pipettes in every 2 microliters. If we want a 0.1, we're going to want one that pipettes has different resolution, and its resolution probably want to be at 0.1, 0.2. So the next little bit is uncertainty. So we've sort of talked about units, we've talked about errors, and actually we get to this idea of uncertainty. So actually we can get a bit of uncertainty in science. It's not a definite, our results are never a definite, so we can get this uncertainty coming about. So uncertainty is officially how confident you can be in a measurement, and so we will always have some sort of realm of uncertainty about it. And so the first thing we talk about is absolute uncertainty, and that is our range of possible real values. So that relates very closely with our resolution. So, for example, if you were measuring a piece of string and it was 10 centimeters and the resolution of your ruler was 0.5 centimeters, there is an element of uncertainty that it could actually be 0.5 centimeters longer or 0.5 centimeters shorter. So this is where this plus minus symbol starts to come in. So we have a plus and a minus together, and that's our uncertainty symbol. So that's meaning, yeah, this piece of string is 10 centimeters plus or minus 0.5 centimeters, depending on if it was 100% correct. So that's our absolute uncertainty, and that is all very closely linked to our resolution. And so that's why we want the highest resolution possible. The higher the resolution we have, the lower that value of uncertainty will be. So that's our absolute uncertainty. That is our range of possible real values. So a range of possible real values from our 10 centimetres would be plus minus 0.5 centimetres. So our potentially the longest that piece of string could be would be 10.5 centimetres. The shortest that piece of string could be would be 9.5 centimetres. Then we can have fractional uncertainty. So fractional uncertainty is your absolute uncertainty that we've already talked about. That's the one that we're sure that there is a definite uncertainty we can have, of, for example, of 0.5 centimetres because of the resolution of our ruler plus our measured value. So absolute uncertainty divided by our measured value will give us our fractional uncertainty. So, for example, we have 0.5 being our absolute uncertainty. Our measured value was 10 centimetres for our piece of string. We would come up with a fractional uncertainty of 1/20th. And that leads us nicely onto percentage uncertainty. That's just our fractional uncertainty written as a percentage. We often quite like to write them as percentages. We can say we're 5% unsure, and they look a little bit nicer. People prefer percentages to fractions, and that's basically all the differences between them. So we do 1/20th, times it by 100, and that will give us 5% uncertainty. So our uncertainty for that measurement would be 5%. Another way we can show uncertainty is error bars and graphs. So these may be something that you've maybe seen before. This is a really silly example to demonstrate it, but someone's got Wonka bars from Charlie and the Chocolate Factory, the sales of them, and during different periods of the year. So error bars basically, the size of them is the error size is proportional to the error bar size. So for example, if we'd had an error bar on this one that was here to there, I mean, I've gone off the graph, so that's not really that helpful, but here to there. And then we had an error bar here, this size. The size of the error bar that you can see is proportional to the size of the error. Okay, so this one here would mean we had a larger range of results, which means we had a larger amount of errors than this one because we have been more precise. Our results are more precise, so we have less of a range of results. And so basically what error bars are is it will mark on our highest result for that reading. So for example, and then our lowest result, and then we have our average in the middle, and then it shows us everything in between. So yeah, the bigger the error bar, the bigger the amount of errors that have taken place. So we've got a bigger range of results, which means we've been less precise. So the smaller the error bar, the more precision has taken place. So systematic errors can be found if theory and results do not match for when the independent variable is 0. So what we mean by that is if our independent variable should start at 0 and our results don't start at 0, let's say our results start here for some reason. So you can plot expected results against your measured results. So if we plotted our expected results and we're expecting it to start from there and go to here, but actually for some reason we're starting at 20 and we might be going up here and then there, we can know straight away that there's a systematic error because that should have started at 0. For whatever reason, whatever we're measuring, it started at 0. If our measurements are not starting at 0 and we are expecting them to start at 0, we know straight away that there's been a systematic error. But it's again relying on the fact that we need to know what our expected results are. Error bars as well can be used to find our steepest and shallowest possible lines of best fit. So if you're doing a line of best fit, you can do them across the average, but you can also do the steepest and shallowest. So our steepest would be G1, so gradient 1, and shallowest would be gradient 2. And what we can do with that is we can do this little equation. So we can do our G1, so our steepest and our shallowest added together, dividing it by 2, plus or minus them taken away from each other divided by 2. So we can find these possible lines of best fit again. So that's quite an interesting thing that we'll come back to for looking at them. So we can do a lot with error bars. So it's more than just showing the range of them. We can find these different gradients as well. And the last little bit on this part is estimation. So estimation comes in a lot, and we talk about estimation generally at this point now as orders of magnitude. So it's the power of 10 that most closely approximates it. So for example, if we were talking about the size, the height of a human, the power of 10 that it would be closest to, if we're talking about the size of a house, you would say it was closest to 10 meters rather than 100 meters. And so that's what the power of 10 estimation idea is. So that is that part. And then I'm going to start off the next topic, because that one's sort of a bit of a theoretical experiment sort of basis. And then the actual first topic, though it's officially topic 2, is particles and radiation. And it's a really nice topic to start on with doing chemistry as well, because lots of it's a repeat. So we sort of going over stuff that we've already looked at, but actually we're looking at them from a physics angle now. So we might focus in, and we focus in a bit more maybe on electrostatic relationships of the particles, and some of the more maths behind it. So it's quite a nice little area to start with. So constituents of the atom. I'll just have a quick talk through this. This is stuff that we have already covered multiple times now. So our atoms, as you know, consist of protons and neutrons. When we combine them two together, we call them the nucleons. So obviously we can have our proton number, or we can have our nucleon number. And also we have our electrons. So our nucleons are in the nucleus, and our electrons, as you know, are in orbitals around the outside of the electron. So the proton and electrons have equal and opposite charges, as we know. So our overall charge of our atom is 0, because our proton has a positive, our protons have positive charges, but our nucleus has a positive charge overall. The orbitals have a negative charge overall, so they cancel each other out. So we're going to have a look in a minute in a bit more detail. This specific charge is the ratio of charge to mass. So we're going to look at that in a bit more detail in a second, so don't worry about that too much. But so yeah, so subatomic particles, they have their charges. What we actually do start to look at as well is the radius of some of these subatomic particles. I'm just going to add them onto this slide now. So we start to look at them even in a bit more detail when we get to physics. So our atom's radius as a whole, so I'm just going to write our atom's radius at the top, is 1 times 10 to the minus 10 meters. And again, we're using meters, we're talking about, we're using that base unit. And this is where it can vary a little bit from chemistry, because in chemistry we can regularly use things like AMUs in different units, so that's something to bear in mind. So yeah, our proton, so that's our atom's radius, and our nucleus radius, I'm just going to pop in our nucleus radius, is 1/10,000th of the whole of the radius of the atom. So our protons, they have their relative mass of 1, their relative charge of 1. Their charge is going to be in, as we've already spoken about the base units, is going to be in coulombs. So 1.6 times 10 to the minus 19. And their mass is going to be in kilograms, because that is our base unit. Neutrons, they have a relative mass of 1. They have a relative charge of 0. And they have a mass of 1.6 times 10 to the minus 27 kilograms. And then finally, our electrons. So our electrons have a relative mass of 0.00005. So officially 9.11 times 10 to the minus 33 kilograms. We start to use this as a relative mass rather than saying 0, because obviously the electron exists, so it does have a mass. So we start to bring this in a little bit more. Our relative charge is -1, so this time it's -1.6 times 10 to the minus 19 coulombs. And we've got our outlines of where protons, electrons, and neutrons are. So clear the drawings. Let's move this on. So specific charge, that's something that we haven't really spoken about yet. So specific charge. So specific charge is that ratio of charge to mass. And we have spoken about charge ratio mass before. And so specific charge is measured in coulombs per kilogram. So charge is measured in coulombs and our mass is measured in kilograms. So for example, I'm going to talk you through an example of a specific charge. We could look at a sodium ion. So sodium ion, we have a mass of 23 proton masses. So that is 3.85 times 10 to the minus 26 kilograms. And it has a +1 electron charge, so 1.60 times 10 to the minus 19 coulombs. So to work out its specific charge, we would take its charge and divide it by its mass. So we would do that and we would end up with 4.16 times 10 to the power 6 coulombs per kilogram. And so you can get asked to work out the specific charge of ions or atoms or even isotopes occasionally. So isotopes, leading us on very nicely. We've done quite a bit of isotopes. So isotopes, as we know, have different number of neutrons for atoms of the same elements. So here we're going to look at one in a bit more detail. We're going to look at hydrogen because actually once isotopes were discovered in chemistry and they went through the mass spec and we're like, oh yeah, isotopes exist. They've actually been used a lot in physics because actually, whilst chemically we've spoken about isotopes, chemically there's not actually much difference in them. In physics, we've actually been able to use some of their differences. There is benefits to them being isotopes. There is benefits to them having this different number of neutrons. So we've been able to sort of use that advantage in physics a bit more. So hydrogen, and we have 3 isotopes of hydrogen. So we have protium, deuterium, and tritium. Again, we have these prefixes that start to suggest 1, 2, 3. So protium obviously has 1 proton and no neutrons. 99, I'm going to add this on actually. 99.98% of hydrogen is made up of the protium isotope. So that is the most abundant hydrogen isotope. And we use this for fuel cells and in the production of plastics. So protium is used for those things. Deuterium consists of 0.02% of all our hydrogen isotopes. And it has 1 proton and it has 1 neutron. And we use this for nuclear fusion. So when we talk about nuclear fusion and we go on to more nuclear physics, deuterium's used there. And then finally, we have tritium. Now, tritium, we're just going to go with rare. Because obviously 99.98 plus 0.02% would make 100%. Tritium does exist, but it's really, really rare. It has 1 proton and 2 neutrons. When it does occur, we use it in thermonuclear fusion weapons. We don't really need to know too much about them right now, but it does have a specific job that we use it for. But yeah, tritium is really, really rare. And we're going to use this new word now called a nuclide. This is something that you can use in chemistry, but it is more of a physics word. So nuclide. So a nuclide is a specific nucleus that contains a certain amount of protons and neutrons. So basically, it's a name that we use for a specific isotope. So we say this is a nuclide. So obviously we have our mass number, our atomic number, A, Z, staying exactly the same, our symbol for our atom, and then a nuclide is the specific nucleus. So actually we're just talking about a specific isotope here. So nuclide, so we could say a nuclide of carbon, a nuclide of nitrogen. So for example, on the screen we've got a nuclide of carbon 12 nuclide. What we are going to get onto is carbon 14. So normally we talk about carbon 12 loads, and we talk about carbon 12 nuclide and how we measure everything against carbon 12 because it's one of the most abundant, it is the most abundant isotope on the planet. But actually, we're going to have a look at carbon 14 now, which is a little bit more exciting. So carbon 14, we use it for carbon dating. So carbon dating is something you've probably heard about. So we use carbon 14, so we use nuclide carbon 14. We just use that word again interchangeably with isotopes because all living organisms have the same amount of carbon 14 atoms as a percentage of all their carbon isotopes. So every single living organism has the same amount of carbon 14 atoms as a percentage of all their carbon isotopes. So what happens is carbon 14 is radioactive. So when an organism dies, it will decay over a known half-life. So that carbon 14 won't get replaced once that organism has died. So what we can do is we can look at the emissions of that carbon 14 from whatever we're looking at, whatever fossil we're looking at, to work out its age. So the older the fossil, the fewer carbon 14 isotopes it will contain, so therefore less radiation will be emitted. So that's really the cool thing about carbon 14 is it's radioactive, so we can use this half-life idea of aging different fossils. Carbon 14 is found even in living organisms, but it comes into play and it is interesting once an organism has died, because then that number reduces. When they're living, it stays the same, and then obviously once the organism has died, then it starts to decay via its half-lives. A little bit more information about carbon dating then. So it's working on that proportion of carbon 12 to carbon 14 in the atmosphere being stable. So there is an issue with carbon dating. We're assuming that the proportion of carbon 12 in the atmosphere and the proportion of carbon 14 are going to stay exactly the same. And that is because carbon 14, where we get carbon 14 from, is cosmic rays. And these cosmic rays interact with nitrogen in the atmosphere. So we've gone with, well, that's going to roughly stay the same. There's probably not going to be too much change. So this proportion of carbon 12 to carbon 14 in the atmosphere is going to stay the same. However, fossil fuels and different things that we have created as humans can affect this. So fossil fuels contain almost no carbon 14 due to their incredible age. So we know that they were living organisms. We know that they would have contained carbon 14, but they are so old that they contain almost no carbon 14. So the carbon dioxide produced by burning these fuels skews the proportion of carbon 12 to carbon 14 in the atmosphere. We're pumping more carbon 12 into the atmosphere than they would normally be, because we're burning up these dead organisms that would have had carbon 14 in them, but it's all decayed already. So we have this skew. Open air nuclear bomb testing also affects this proportion in the opposite way, and it drastically increases the amount of carbon 14 in the atmosphere. So open air nuclear bombs, they have carbon 14 inside them, with it being a radioactive isotope. And so when that happens, that increases the amount of carbon 14 in the atmosphere. Carbon 14, so other than carbon dating, we can use it for a few other things. We can use it in nuclear fission. So we can use it to produce large amounts of energy via a chain reaction, which uses radioactive isotopes of uranium or plutonium as fuel. Molecular labeling is really interesting. This is used a lot in chemistry and biology. And what we do is we use radioactive isotopes, such as carbon-14, to track mechanisms of specific molecules. So what we will do is a regular atom is swapped out for an isotope, or we can actually add on this radioactive isotope. It behaves exactly the same way as the original atom because the proton number is the same. And what happens is this isotope is now detectable due to its differing number of neutrons. And because it's got differing number of neutrons, because it's radioactive, we can then track it. So this is used a lot, even in people when they're alive. So often we might want to track if there's blockages in people's bodies or how the blood's flowing, and people can basically take a drink that has small amounts of radioactive isotopes in them, and then we can track them and see where in the body they're moving to, because they will obviously let out radioactive emissions. So we make sure that they are the right sort of radioactive isotopes, and they're not going to cause permanent lasting damage to somebody, but they're really quite clever for showing the inner workings of the human body. And we can also use it for transition electron microscopy. So this is a form of microscopy which passes a beam of electrons through a sample, forming an image. He's basically made almost like a 3D image. So rather than microscopy, which would usually give you a flat image, we get this more of a 3D image, and we basically beam these electrons onto this image, and we get this really nice, really clear image. And that's something we'll look at a bit more actually in all three sciences, and then it produces these really fantastic scientific images, but they're actually really, really beautiful as well. So we're just going to have a little look at strong nuclear forces. And this is that stability of a nucleus again, and that quantum theory we talked about. So our nucleus, it has a positive charge, it's got protons and neutrons in there. This idea of strong nuclear force, so without a strong nuclear force, our nucleus would break apart. And that's because of the electrostatic repulsion between the protons. So when we were talking about yesterday about electrons repelling each other because they are of the same charge, so they're going to have some effects of repulsion, the same thing happens in the nucleus. The protons, both positively charged, they can start to repel each other. So we have this nuclear force, and this nuclear force basically keeps our nucleus together and overrides this electrostatic repulsion between the protons. It is a short range force, so it's only significant over a really short distance. So two protons that are separated by more than 3 FM nuclear force is too weak to overcome that electrostatic repulsion. So it only works over a really short range, which is important because otherwise it could then have an impact further onto how it reacts with the electrons further into the atom. So here is a graph showing this nuclear force. So it shows that it's a short range force. Anything further than 3 FM, it's too weak to overcome the electrostatic repulsion. But it's also repulsive at very short distances. So whilst we're saying, oh yeah, it's attractive, it keeps everything together, we go back to that idea of actually, when we're saying that quantum theory, a whole atom should collapse in on itself. The pull from the electrons to the protons should pull it in, and then it would collapse in. And actually, if we've got an attractive force happening in our nucleus and it's keeping all those protons together, counteracting the repulsion, it would actually cave in on itself again. It would collapse into a point. So at short distances, this nuclear force, this nuclear force is also able to be repulsive. So it must be repulsive at some more distances of things like 0.5 FM. So it's really, really clever on how it's working. So it's actually able to be repulsive and attractive at the same time. So it's just sort of like an extra layer then really to the information that we've learned in chemistry about the electron shells and the electron levels, and that's good information to know. I haven't gone over all of that here because there's sort of no point. We've been through it, we understand it really well, so there's no point really going over that. But yeah, I find this really interesting, and the way that it works, the forces. So this one force, this strong nuclear force, is able to be over short range attractive to overcome those electrostatic repulsions, but actually at a really, really short distance, it's able to be repulsive to stop that caving in of the nucleus. So yeah, what we will do on Monday is, let me stop recording this.